Until the beginning of colonization in the 15th century, most of the world did not exhibit this characteristic. It was then that the Rh factor, positive or negative, was identified, with Rh positive being predominant in the human population. For a long time, type O blood was seen merely as a routine clinical detail, a bureaucratic note in medical records and donation campaigns. However, behind this simple classification lies an enigma that resists easy explanations. The distribution of type O blood around the planet reveals patterns that don't perfectly fit known models, leading researchers to reconsider old certainties about this unusual genetic signature. In medicine, blood typing is usually presented directly: A, B, AB, or O. Among these, type O stands out for not having A or B antigens on its red blood cells, making it the universal donor. This neutrality drastically reduces the risk of immune rejection and makes it a reliable donor for doctors in emergency situations when there is no time for compatibility testing. Over the decades, this characteristic has saved countless lives. Outside the hospital setting, however, this same neutrality hides something intriguing. Studies in population genetics have shown that in large regions of the Americas, from Alaska to the Amazon, type O blood is not only common, but dominant. In some indigenous populations, it is practically exclusive. Amazonian tribes, Andean villages, and Arctic communities exhibit successive generations with almost total uniformity of this single blood type. The initial explanation seemed simple: population isolation and genetic drift. In small, isolated groups, certain alleles can randomly become predominant, while others disappear. But as the data accumulates, there is a growing sense that this answer may not be sufficient to fully explain why type O blood simply refused to disappear. A related concept, known as the founder effect, suggests that if a small group of people with similar genetic characteristics gives rise to a new population, those traits will remain predominant among their descendants. These ideas seem to fit well with the story of type O dominance in remote communities. After all, if the original settlers of these regions had type O blood and remained isolated from outside populations, it wouldn't be hard to imagine how this trait could persist for centuries. But as more genetic data began to arrive from across the Americas, the chance theory began to lose traction. The pattern was too consistent. It wasn't just one or two isolated groups showing a high frequency of type O, it was practically everyone. And the uniformity went beyond what would be expected from random genetic deviation alone. Even in populations that had some contact with outside groups, the dominance of type O remained strong. In some cases, other blood types were not only rare, they were completely absent. This degree of purity raised difficult questions. If genetic deviation were the only factor, it would be reasonable to expect at least some pockets of variation, traces of other blood types mixed into the genetic makeup. Instead, the data indicated something different, as if an intentional filter had been applied at some remote point in the past, preserving type O while types A and B were completely erased from the genetic record of entire regions. For years, this implication was quietly ignored. It was more comfortable to adhere to traditional explanations than to admit the possibility of a deeper and more disturbing cause. Over time, however, this stance became untenable. As archaeologists began unearthing human remains from very ancient periods, and geneticists developed tools capable of extracting DNA sequences from bones thousands of years old, the story of type O ultradominance only deepened the enigma. The scale and uniformity of the phenomenon suggested that it was not a historical accident. On the contrary, it seemed like a signal, pointing to much older and much stranger origins. It was then that researchers decided to look even further back, investigating not only current populations, but humanity's own migrations. The more they traced the path of Type O, the more the familiar narrative about how humans arrived in the Americas began to crumble under the weight of new evidence. The more scientists investigated the origin of Type O, the more the classic story of ancient migrations began to crack. Cracks in the ice bridge theory. For decades, anthropology textbooks have told a single, organized story about the arrival of the first humans in the Americas. Approximately 15,000 to 20,000 years ago, during the last ice age, sea levels were much lower than they are today. The Bering Strait, now a narrow strip of icy water between Siberia and Alaska, was once a vast land bridge known as Beringia. According to traditional theory, small groups of hunter-gatherers crossed this frozen expanse, following herds of animals to a new and uninhabited world. Isolated from their Asian homeland by rising seas and melting ice, these pioneers became the ancestors of all the indigenous peoples of North and South America. The genetic picture seemed simple. If this migration involved only a small number of people, perhaps a few hundred, they would have taken with them only a portion of the genetic diversity present in the original Siberian population. Over thousands of years, isolation would amplify these initial traits, explaining why certain genetic markers became dominant among indigenous peoples. Following this logic, the overwhelming prevalence of Type O blood in the Americas could be explained if the founding migrants simply possessed, mostly or entirely, Type O alleles. It was an elegant and self-contained theory, compatible with the tools and evidence available at the time. But then came the revolution in ancient DNA research. As scientists learned to extract and analyze genetic material from skeletal remains thousands of years old, they began to closely examine the blood type alleles present in ancient Siberian peoples, precisely those who, according to theory, would have crossed the land bridge to the Americas. The results did not fit the traditional narrative. Far from being genetically uniform, these ancient Siberians exhibited a wide variety of blood types: Type A, Type B, Type O. All were present in significant numbers. This was not a small, genetically limited population dominated by Type O. It was a rich and diverse group, much like what we see today in Europe, Asia, and Africa. The implication was clear. If the first Americans came from this population, they would have brought with them the same diversity of blood types when crossing the land bridge. And that's where the problem began to reveal itself. If multiple blood types arrived in the Americas, why don't they remain present in present-day indigenous populations? Where did the A and B alleles go? Their complete or near-complete disappearance is not what we would expect from a simple random genetic drift. Drift can reduce diversity over time, but it rarely eliminates important alleles entirely, especially on a continental scale. Instead, the genetic evidence pointed to something far more dramatic, often described as a genetic bottleneck, a collapse in blood type diversity after humans arrived in the Americas. The A and B alleles not only declined, but in many regions they became extinct, leaving Type O as the sole survivor. This was not a slow, random process. It was sudden, decisive, and essentially synchronized. Such a pattern raised unsettling possibilities. Had blood group AB been eliminated by some calamity that struck the first populations? Had a pathogen emerged that selectively affected these blood types, conferring an evolutionary advantage to Type O? Or had some other selective force, still unknown, acted on the population, reshaping the genetic landscape in just a few generations? Whatever the answer, the clean and comfortable story of migration no longer held up. The disappearance of blood types A and B in the Americas was not the kind of genetic change that could be easily explained by geography, climate, or chance. It was the signature of an event, a powerful filter through which only one blood type passed intact. Evidence was beginning to suggest that the journey to the Americas was not just a migration, but the beginning of a test of survival, whose scars are still visible in our DNA. And what scientists found in ancient human remains indicated that this disappearance was not slow. It was deliberate. The bloodlines that disappeared. In 2025, the mystery of type O dominance took an abrupt and disturbing turn. A coalition of geneticists from Canada, Brazil, and South Korea launched an ambitious project to map the history of blood type alleles in the Americas using ancient DNA. The approach was unprecedented in its scale, sequencing genetic material extracted from skeletal remains spanning thousands of years, from Alaska to Patagonia. The goal was simple: to track the presence of blood type A, B, and O alleles over time and across geography to understand how current patterns formed. The results, however, were far from straightforward. The earliest human remains found in Alaska, close in both time and place to the first crossing from Siberia, told an unexpected story. These ancient Alaskan inhabitants possessed not only type O blood, but also faint traces of type A. This alone represented a decisive advance. This proved that the first people to arrive in the Americas actually brought more than one blood type with them. The old theory that only type O blood crossed the land bridge was no longer tenable. However, as researchers analyzed remains found further south, traces of type A blood began to disappear. In skeletal remains from the Central Plains of North America, type A was absent. In samples from Mesoamerica, it had disappeared. When they arrived at the remains of the first Andean peoples of South America, there was no sign of either types A or B. For thousands of kilometers, the story repeated itself. Only type O remained. This abrupt disappearance contradicted the slow and irregular patterns expected from random genetic drift. It seemed less like a gradual fading and more like a purge. Something, or someone, had removed types A and B from the gene pool with ruthless efficiency. The researchers began to speculate. Could a catastrophic epidemic have swept through the Americas, exploiting specific vulnerabilities of blood types A and B? Certain pathogens are known to bind to blood group antigens, using them as entry points into the body's cells. If such an infectious agent targeted antigens A or B, those with type O blood would have enjoyed a survival advantage. Other hypotheses were equally grim. Perhaps a previously unknown virus or bacterium, now extinct, devastated ancient populations, sparing only those with the genetic makeup to resist. Others have suggested that environmental factors may have played a role, with extreme climate change or nutritional stresses interacting with the immune system in ways we do not yet fully understand. Still, all these scenarios pointed to the same conclusion. Something highly selective had been at play. The most revealing clue didn't come from the main body of the study, but from a single sentence easily overlooked in its abstract. The data indicate a directional selection event, possibly of immunological origin, favoring type O alleles over all others. In simpler terms, this meant that the changes in the distribution of blood types were not random. They followed a direction, a powerful push from evolutionary forces towards type O, and these forces were almost certainly linked to immunity and survival. It was a disturbing finding. She suggested that the near-total dominance of type O blood in so many indigenous populations was not merely a result of geography or chance. It was the mark of a survival test that swept across the Americas in a deep prehistory, a test so severe that it left only one blood type standing in vast regions of the continent. If that were true, then blood type O was more than a medical convenience. It was the genetic legacy of those who survived when others perished, the very blueprint for survival. If type O blood survived where others did not, what hidden advantage did it carry in its own code? The immunological gap. When scientists began to investigate what made type O blood so dominant in certain populations, the answers began to point to something beyond simple genetic chance. Patterns began to emerge in global health data. Patterns suggested a silent resilience hidden in plain sight. People with type O blood across different continents and environments consistently showed lower rates of severe forms of some of history's most dangerous diseases. Cholera, malaria, dengue fever, and even certain variants of severe respiratory infections seem to take a slightly lower toll on those with type O blood. This does not mean that individuals with blood type O were immune. They could still contract these diseases, sometimes fatally. But when survival odds were analyzed in large populations, type O seemed to have an advantage. It was a subtle advantage, not enough to protect everyone, but enough so that, over generations, more type O carriers survived outbreaks. The more researchers compared modern survival rates with historical disease patterns, the clearer it became that this silent advantage could have been crucial in the distant past, when medical intervention did not exist. The most striking evidence came from the highlands of the Andes Mountains in South America. There, in some of the harshest living conditions on the planet, communities not only endured for centuries, but thrived. The air is thin, the sun's ultraviolet radiation is intense, and exposure to infectious diseases has been constant for generations. Still, many of these communities exhibit surprising levels of long-term survival. When geneticists analyzed their blood, they found something remarkable: an unusually high prevalence of a genetic variant known as IL-17, variant 52C. This small alteration in the immune system's design affects how the body responds to infections. In many serious illnesses, death is not caused directly by the pathogen, but by the immune system's own overreaction, a cytokine storm that causes widespread tissue damage. The IL-1752C variant appears to function as a biological thermostat, keeping the immune response strong enough to fight infection, but controlled enough to prevent self-destruction. What made this discovery even more significant was its strong association with type O blood. In these Andean populations, type O and the IL-1752C variant appeared together with a frequency far greater than chance would allow. Over eons, this suggested that something had been continually testing these populations, eliminating those who could not achieve the proper balance in their immune systems. Surviving with type O blood and the IL-1752C variant, these individuals were more likely to resist and thus pass on their genes to subsequent generations. The probability of this occurring by mere chance is small. It involved a long and arduous history of selective pressure over eons, with repeated struggles for survival, possibly against pathogens whose existence we are now unaware of. Whatever the reason, this pushed human biology to a limit, favoring individuals with type O blood associated with this immunological variant, who remained silently resilient under its influence. These results were not widely publicized. They remained confined to specialized journals, accessible primarily to experts. There were no headlines on the evening news announcing that a particular blood type might carry echoes of ancient evidence of survival. Yet, for those paying attention, the evidence was mounting. Type O was not just a universal donor. It could be the living memory of a time when only the most adaptable immune systems survived. And as scientists continued to search for more clues, one discovery stood out above the rest. It came from the frozen soil of Siberia, preserved for more than 12,000 years, and carried with it a mutation unlike anything researchers had ever seen before. So, frozen deep in Siberia, an ancient skull revealed an unprecedented mutation, the Lake Ball marker, In the remote, icy regions near Lake Bull in Siberia, archaeologists unearthed a discovery that would further deepen the mystery surrounding type O blood. Beneath layers of ice and soil, preserved in the permafrost for over 12,000 years, lay the skull of an ancient human. The conditions had kept the remains in an extraordinary state, and geneticists knew this was a rare opportunity, one that could offer a direct glimpse into both the distant past and the future of human lineages. When the DNA was sequenced, the researchers found something extraordinary. The individual carried a rare genetic marker in the ABO blood group gene set. It wasn't a common mutation, but a deletion. An entire section of the genetic code was missing. Scientists have named it the O delta variant. 1. Deletion mutations of this type are much less common than point changes in DNA and generally indicate that extreme evolutionary pressure forced the organism to adapt drastically. What made this discovery even more fascinating was its uniqueness. The O delta variant 1 was not present in European DNA. It did not appear in African or East Asian populations. Even within Siberia itself, it was absent in most ancient samples. The only place where this genetic signature emerged was among certain indigenous groups of North America, establishing a clear link between the ancestral Siberian individual and the descendants who would later populate the Americas. For geneticists, this was like finding a unique fingerprint spanning millennia. The deletion appeared to have a specific function, possibly influencing how the immune system recognized threats. This suggested that, at some point in prehistory, this lineage faced an intense, perhaps life-or-death challenge that reshaped its genetic code. The implication was staggering. This was no random variation. It was the residue of a biological battle. Something, whether an environmental toxin, a long-forgotten disease, or a catastrophe that triggered the immune system, left such a profound mark on the biology of that population that it altered its genetic structure in a way that is still detectable today. And it wasn't a general adaptation of the human species. It was something specific, belonging to a single bloodline that survived while others disappeared. Even more intriguing was how closely the O delta variant 1 was associated with type O blood. In modern indigenous groups where this variant appeared, type O was overwhelmingly dominant, sometimes to the point of exclusivity. It wasn't just about those who had crossed the Bering Land Bridge. It was about those who survived long enough to pass on their genes. For anthropologists, this discovery added a sense of urgency to the mystery. If the same genetic marker could be traced from an individual who lived in Siberia during the Ice Age to communities living thousands of kilometers away, it meant we were facing an unbroken chain of survival. Throughout climate change, migrations, and catastrophic events, this trait had persisted. Yet, however significant it was, it represented only one piece of the puzzle. It indicated that there had been intense pressure on early human populations, and that this pressure had produced lasting genetic changes. But it didn't say exactly what the threat had been, nor why, in particular, type O blood seemed to carry so many of these survival markers. That answer would come from a completely different discovery, not in the frozen tundra, but in a modern DNA sample submitted in 2025. This case would reveal something no one expected: a possible built-in defense switch hidden deep within the human genome. In 2025, however, a single man's DNA sample revealed something even stranger: a possible hidden defense mechanism. The Crawford revelation. In early 2025, a discrete entry in a genetic database triggered a chain of events that no one could have predicted. It all began with Thomas Crawford, a resident of Blackfeet Nation in Montana, who agreed to participate in a large-scale ancestral mapping project. Like many volunteers, he expected the results to simply confirm his heritage and offer a detailed portrait of his lineage. But when his DNA sample was analyzed, it revealed something far beyond routine ancestry. Crawford carried the O delta variant. 1. A rare deletion mutation first identified in a 12,000-year-old skull found near Lake Bull. That alone made their sample remarkable, linking it to one of the oldest and most unique genetic signatures known in the Americas. But it was what appeared alongside O delta that left the researchers completely baffled. In the DNA sequence surrounding the human leukocyte antigen region, HLA, a critical core in controlling immune responses, they found an unusual fragment of genetic code. For decades, this segment had been discarded as junk DNA, a term used for sequences with no known function. However, when advanced immune modeling simulations were applied, the fragment behaved unexpectedly. Under normal conditions, it remained inactive, silent in the genome. But when the model simulated extreme biological stress, scenarios involving multi-organ infection combined with oxygen deprivation, the sequence was activated, not partially, nor weakly, but like a light turned on in a dark room. Once activated, it appeared to regulate a cascade of immune defenses, stabilizing inflammation and protecting vital organs from collapse. In essence, it acted as an emergency survival trigger, activated only in the most life-threatening situations. The researchers had never seen anything like it. It was as if an ancestral backup system had been written into the genetic code, a hidden defense that may have been the difference between life and death during catastrophic outbreaks. And yet, this mysterious sequence did not appear in all human genomes. It was disproportionately prevalent among individuals with type O blood in certain indigenous populations, often alongside the O delta variant. 1. While the team debated the implications, an even stranger twist emerged. According to a whistleblower within the Human Genome Archive, a vast international repository of genetic data, this same sequence had already been identified before. The source claimed it was first flagged in 1998 during the early stages of the Human Genome Project. At the time, its function was unknown, but its unusual structure had attracted attention. Then, inexplicably, the information disappeared from public datasets. The whistleblower claimed it was deliberately buried, never discussed in published research, and excluded from major genetic databases. Why? No one outside the inner circle seemed to know. Perhaps the discovery was considered too speculative at the time. Or perhaps the implications of some human lineages carrying a unique, possibly even engineered, survival mechanism were seen as too controversial to publicize. Whatever the reason, it took almost 30 years for the fragment to reappear, this time directly linked to the mystery of type O blood. For the scientists working on the Crawford case, the discovery was both exciting and frustrating. They had a tangible link between type O blood, ancient survival events, and a hidden genetic defense system, but no clear understanding of how it had arisen or why it appeared only in certain populations. One thing, however, was certain. The trail of type O history was no longer confined to the Americas. New genetic comparisons suggested connections far beyond, crossing the Pacific Ocean and linking survival codes between peoples separated by thousands of kilometers of open sea. And when scientists traced type O blood beyond the Americas, the path led them across the Pacific to a striking connection: the Polynesian Passage. The final piece of the puzzle came from one of the most remote inhabited places on Earth, Rapa Nui, better known as Easter Island. Located thousands of kilometers from the nearest continent, its isolation has long fascinated historians and archaeologists. In 2025, excavations on the island unearthed skeletal remains dating back approximately 900 years. These bones, preserved in the island's volcanic soil, were sent to genetic laboratories for detailed analysis. The results surprised the researchers. The remains not only showed an overwhelming prevalence of type O blood, but also carried the IL-1752C variant, the same immune-modulating genetic variant found in Andean highland populations in South America, thousands of kilometers away. This was no trivial coincidence. The chances of the same rare immune variant arising in both locations purely by chance were astronomically low. The most plausible explanation was direct genetic exchange, which, in turn, suggested contact between Polynesian navigators and South American peoples centuries before the arrival of Europeans. For years, the idea of pre-Columbian contact across the Pacific was debated in academic circles. The evidence consisted mainly of similarities in crops, artifacts, and oral traditions. Now, the DNA itself was speaking. It wasn't just a cultural exchange, it was a biological exchange, encoded in the blood. If Polynesian navigators truly reached South America, they may have brought with them not only goods and ideas, but also the very genetic tools for survival against deadly diseases. Meanwhile, another line of research was taking shape high in the Andes. Archaeologists studying the teeth of ancient Andean individuals found tiny traces of antibodies attached to the tooth enamel. Microscopic records of immunological battles fought throughout their lives. Some of these antibodies were directed against pathogens that modern science has never encountered. They likely belonged to diseases now extinct, long since erased from human experience. What made the discovery even more unsettling was who carried these immunological markers. Almost all cases came from individuals with type O blood. The other blood types were almost absent from the archaeological record in these contexts, not because they were neglected, but because those who possessed them may not have survived the waves of disease that swept through ancient populations. This painted a grim, yet revealing picture. During periods of severe epidemics or extreme environmental stress, blood type O was not only the most common, it may have been the only one capable of surviving. Whether due to greater immunity, genetic resilience, or simple adaptability, blood type O has repeatedly survived where others have disappeared. With each crisis, it has become more prevalent among the survivors, consolidating its place in the genetic heritage of these populations. The Polynesian connection has further amplified this narrative, indicating that its dissemination occurred not only through land migrations, but also through long ocean crossings, undertaken by people on journeys of exploration, trade, or alliance. Over time, what seemed like just a medical curiosity revealed itself to be a profound story of human contact, catastrophes, and resilience. Traces of type O blood appear from the Amazon to Easter Island, from the Andes to the Siberian permafrost. Each piece of evidence reinforces that it is more than a simple clinical classification. It is the result of successive tests of survival, biologically imprinted on those who carry it. This trajectory suggests a legacy of near extinction and extreme adaptation, a persistent code that has weathered collapses and ensured continuity. Type O blood may not only be the universal donor, but one of the clearest signs of human survival itself. The question that remains is what we do when we discover that our blood may carry such a profound history.